All right, good morning, everybody. Give me 1 second here, sorry. So today we're gonna keep working on genetics. That's kind of our theme all week. And we're gonna keep going today. But just before we get started, I do kind of wanna give a reminder. I put in the zeros for last week on Monday, which was yesterday. But please just be sure to go in and double check. A lot of people have been, for whatever reason, there's a tech issue on Unified Classroom where some people are turning in assignments that are completely blank. And I'm not really sure why it's happening. It might be, I'm not sure what it looks like on the student end. It might be that you're not saving before you're submitting your work on Unified Classroom. But I'm not 100% sure what's happening. So please, please, please double check your grades. If this happened to you, if I got a blank assignment from you, it should have a 0 and then a little comment from me next to it that says, hey, can you please submit this again? For whatever reason, it was blank on my end. But just please be sure that you're saving and double checking what you're actually submitting after you've done it, because yesterday it was like 6 or 7 times that this happened. And I'm not entirely sure what's going on your guys' end, because I am getting plenty of other assignments on Unified Classroom that are coming in normally. But I think that my understanding is you have to press save your work before you can submit. If you just submit, it will submit something that is blank. So please double check that. Please go back in on Unified Classroom and check your grades. If there's something that's a 0, just send it to me again. I'm not going to take off points, but just be aware that that is happening and just make sure you're on top of yourself. I really don't want to get to the end of summer school and have everybody say, well, I submitted everything, because if it's blank, I really can't grade it and give you credit for it. If this is an issue for you, and if this has been happening for you, and you're worried about it, honestly, the best way is, if Unified Classroom isn't working for you, if you're not sure how to submit your assignments right, or if you've been having a lot of tech issues, just email me your assignments. You can just write your answers straight in the email and send it. That's also fine. But just keep in mind, this is happening, and I've been seeing it a lot. So just make sure that you don't have zeros on Unified Classroom that you don't know about, and make sure that you're submitting your assignments correctly. All right. So today's topic is going to be transcription and translation. We are going to be building off of that DNA and RNA lesson from yesterday. So if you didn't do that lesson yet, I really strongly recommend that you go back and do that one before you try and do this one, because these two lessons sort of build on each other. So yesterday we were learning about DNA, and how DNA has a code in it. That code is super important. It's what makes you you, and it's what makes your body function. Today we're going to be learning about the other couple things that happen. So it's not just your DNA that's involved. Your DNA can make something called RNA. And that RNA then goes on later to make a protein, which sometimes you might see written as a polypeptide. So today our focus really is this transcription piece and this translation piece, where we're going from DNA to RNA and RNA to protein. And that's really what we're going to be practicing today. And it can be a little tricky. So if you get stuck, please be sure to reach out to me. This is just review. You guys don't need to write this down yet. Yesterday we did learn a little bit about the differences between DNA and RNA. The two differences that we focused on was that DNA had this letter T, while RNA did not have that letter T. It had a letter U. That's going to be very important today. Another thing that was important was DNA had 2 sides to it. It was double-stranded, right? So if I were to draw a DNA, it would kind of have 2 sides to it, versus RNA would just be like 1/2 of that DNA. It wouldn't have both sides to it. So today, and this is kind of where we start our notes, we're going to be doing 2 things. The first thing we're going to be doing is transcription, and we're going to practice it together. All right, transcription is where you're taking the DNA sequence and we're turning it into RNA. So we're going from DNA to RNA. And we learned a little bit about why this was yesterday, but DNA can only stay in the nucleus of your cell. It can only stay trapped in one spot. So it needs to do transcription. It needs to make RNA to send that message out. We're going to actually be practicing on how to make that message today. So this sequence should be the exact same one that you have on your sheet, all right? And it's very similar to what we were doing yesterday with our DNA sequence. So yesterday we were saying that letters C and G always go together, right? So C makes G if this is an arrow. We can say the opposite. We can say G makes C. If you have a T in DNA, that's going to turn into an A in RNA. But remember, if we have an A in our DNA sequence, instead of it becoming T like we practiced yesterday with DNA, if we're making RNA, that's now going to turn into the letter U. All right, so here, these are my DNA letters. Sorry, my writing is difficult with a mouse, right? And this is what they would turn into on RNA. All right, so it's the same idea of A to T and C to G that we learned yesterday. It's just in this case, anytime there's a T, we're actually just going to write down the letter U instead. So we just have to remember that part. So for example, this might be my DNA right here that I gave you. Our job now, if it's telling us to transcribe it, is going to be to write down what the RNA is. So we have C, right? We know C goes to G. Right, so it would be G. This is the tricky part. We have A. Normally in DNA, A would go to T, right? But we know now, since we're going from DNA to RNA, we actually need a U. This would be G, U, C. Remember, C and G go together. That T is going to go to A, right? Remember, there are still A's in RNA. A, C, C. Right, remember, we have an A again, so we have to use U. U, U, G. My gosh, my handwriting, you guys. G goes to C. A goes to U. T goes to A. All right, so I know that's tricky. If it helps you, you might want to write down this sort of a chart on your paper just to help you out. All right, but you already did this yesterday. You were really good at it yesterday. Just remember, you're writing U's instead of T's. That's the only difference. All right. I'm going to give you guys another sample sequence. This one is not on your paper, but I really strongly recommend just try it for a minute. All right, just think to yourself and pause. All right, and if you're unpaused, this would be your answer. All right, so you're just seeing, you're turning your letters into the ones that go together. All right. And if you have trouble with this, please send me a message or join my office hours. I'm happy to do that for you. All right, so that's transcription. Transcription, we're just going DNA to RNA, and we're making sure that that RNA sequence is correct with our letter U. The second step is translation. So at this point, we've got our DNA. That DNA has created that RNA message. Now that RNA message has to actually do something. So translation, we're taking that RNA sequence, that RNA message, and we're finding the protein that it makes. All right, so we're going, before we were going DNA to RNA, at this point now we're going RNA to protein. We talked a little bit about proteins last week, not too much, but proteins are super important for you just to be alive. You would not be here if you didn't have proteins working. Proteins are doing pretty much all of the important jobs in our body. Proteins are letting your oxygen go through your blood. They're helping your brain function. They're allowing you to digest food. They're what makes your skin color and your hair color what they are. So proteins are being made all of the time, and they're doing a lot of really important jobs. One key thing I want you guys to remember as we're practicing translation is you're always using the RNA sequence. All right. There are going to be a couple questions later on where I'll give you a DNA sequence. but you have to make the RNA sequence first. If you're not using RNA, it's not going to work. All right, so just keep that in mind. So you may have seen this chart before when you guys were taking biology last year, but it is a little bit tricky, so I'm going to use it along with you guys. This sequence here, it's the exact same one you have on your paper, so I'm just going to do the notes section with you. All right, but this is a sequence of RNA. We know it's RNA just looking at our first 3 letters, that it does not have T in it. Right, so we know it's RNA, not DNA. What we're doing is, you've probably noticed I've already broken these up into 3-letter segments. So you always go 3 letters at a time. And for this class, at least, I'm always going to have them separated like this so that it'll help you remember. And what you're doing is, your protein is basically one long chain with lots of little segments. So I think of a protein almost like a big long train, where there's lots of little pieces all connected to make one big train. So we're going to go through each of the pieces, and we're going to find those 3 RNA letters on our chart. And there is kind of a sneaky way to go about this to make it a little faster. You can take the first base, the first letter, and start there. But honestly, you can just look for it, and that's okay. So we're going to look for the letters U, U, U. All right, U, U, U is right here up top left. We're going to write down what it makes. And sometimes there might be more than one RNA sequence that makes the same thing, and that's okay. So RNA makes Phe, P-H-E. So in our answer, that's going to be the first thing that we write down is P-H-E. These are all shortened. Technically, P-H-E stands for something called phenylalanine, which is the piece of the protein, but honestly, all I want is just first 3 letters. Don't go crazy with this. All right. Our next 3 letters are A, A, G. All right, so my first letter here is A, so I kind of know to start down here somewhere. Right, I'm going to look for A, A, G, which is right here. A, A, G makes lyse or lysine. Right, we're going to keep going. So G, G, U. If I look in this first column, G is down here, so I know I'm going to be looking kind of at the bottom of the chart. So G, G, U is here. G, G, U makes glycine, G, L, Y. All right, and so on and so forth. I'm going to do these last couple ones a little bit more quickly. CAG makes GLN. And UAG makes stop. So stop is not actually a piece of the protein. Stop actually means what it says. So stop means, hey, you're done. You made the end of the protein, and it tells your body that that protein is done, and it can stop making it. All right, so that's what my answer would look like on my note paper and what I'm looking for from you guys. All right, so you can try again if you want to. It's up to you. I'll do it very, very quickly if you want to get some more practice that's not on your paper. AAC. Where's my little guy? Ooh, that was supposed to be an A. I'm sorry. ASN, CCU, or CUU, sorry. Leucine. GUA. Right there. And AGG. AGG is ARG. All right, so if you're checking your answers, that is what you're going to end up with. All right, but that's pretty much it for today. All right, I'll show you your assignment very quickly, and then I'll let you guys go. So you guys are going to do a couple little practice problems here. You're going to find the RNA first, and then after you make the RNA, you're going to find the protein. All right. Remember, for that protein part at the end, I have this chart on your paper. You might just need to go to the bottom page in order to use it. You guys are going to just do 3 times of that. All right, it should hopefully be pretty quick. And then we talked a tiny bit about mutations yesterday. We're going to do a little bit more today. There's a short reading here. It's like 1 page, that's it, from National Geographic. And then you're going to answer 4 questions, and that's it. All right. And then after this, we're going to kind of move on tomorrow to a new topic. All right. So try your best. Again, please double check Unified Classroom. Double check that your grades all look good. You don't have any zeros that you're confused about. If you are having issues submitting stuff, talk to me or just email me your assignment that way, whatever makes it work. But just please make sure you guys are on top of that. But otherwise, I'm here for you guys today. If you guys need any help, send me a message however you like.